immune system of our body is comparable to the defense system of a country. Now, defense system of a country has different divisions, like a air force, like a land force, and a navy. And among all this division, what is important is communication. So there should be several ways by which all these divisions of the army of a country can communicate between each other. And they can communicate between radio, walkie-talkies, satellite phone, etc. Also, our immune system should devise a communication strategy such that all the players of the immune system can communicate with each other. Immune system cells communicate with each other either directly going to each other physically, or they communicate by several wireless media. In their case, this wireless mode of communication is via small chemical messengers known as cytokines. So in this video, I would talk about cytokine mode of action, 6 families of the cytokine, signaling mechanism and the function of the cytokine families, and also the clinical use of cytokine. So if you haven't subscribed my channel, why are you waiting for? Please hit that subscribe button and don't forget to like my video. So, communication modes used by a normal cell could be either endocrine, where the cell secretes a messenger molecule, here depicted in green, which flows through the bloodstream and reaches its distant target cell. Other mode of communication could be paracrine, where the cell is not so far away, the target cell is not so far away from the cell signaling the cell which is secreting the signal. So that is paracrine. Another is autocrine, means the cell itself signals a molecule which works upon the cell itself. Now the immune system uses mostly paracrine and autocrine mode of communication systems. Now how it works is there could be inducing stimulus which can allow for activation of specific cytokine genes, and that would ultimately lead to cytokine mediated signaling in the recipient cell or the target cell, and the target cell would have a variety of biological effect due to this cytokine. It would either divide, it can differentiate, or even sometimes it can get apoptosis, it can undergo apoptosis or death. So variety of outcome could occur on cytokine signaling. So let's just breeze through the various functions of cytokine. So cytokines are useful for multiple different purposes in the immune system. For example, lymphocyte migration requires a specific type of cytokine known as chemokines. So lymphocyte cells moves along the chemokine gradient to reach their destination. Also, development of several immune cells, for example, B cell, requires specific cytokine derived signaling. So interleukin 7 and interleukin 7 receptor mediated signaling is very important for VDJ recombination, which is important for a B cell development to occur. Also, T cell differentiation is also important process for an immune cell, and T cell differentiation requires several different cytokines, and several different cytokines guide the differentiation of T cell to several platoons of T cells. And I have several videos on B cell development and role of cytokines in T cell differentiation. So you can watch those videos. Also, several cytokines can give inflammatory responses, such as IL-6, IL-1, etc. that we would be discussing very soon. Also, T cell activation requires several cytokine signaling. Also, in several cases, cell proliferation or proliferation of the immune component cells requires interleukin, or which is one type of cytokine. So that means cytokine is widely used by the immune system for several purposes: for growth and development, for differentiation, for reaching the destination, otherwise migration, and also manifestation of inflammation, specific activation, and cell proliferation. So a wide variety of function is performed by the cytokines. Now here is the 6 cytokine families, namely interleukin 1 family, and its family member are shown here. One of the important most family member of interleukin 1 family is IL-1, which is the prevalent pro-inflammatory cytokine. Second is hematopoietin family. And one of the most important member is interleukin 2, because interleukin 2 is very important for T cell proliferation. Now interferon family is very interesting, and interferon family has interferon alpha, beta, gamma and interferon lambda. Interferon alpha and beta are important and they are pro-inflammatory, whereas interferon gamma gives a protection against viruses. And interleukin 10 or IL-10 is an anti-inflammatory molecule secreted by the T regulatory cells. There is an important family also known as tumor necrosis factor family. So tumor necrosis factor family has TNF alpha, TNF beta, CD40 ligand, FAS, BAF, APRIL, etc. as their family member. Interleukin 17 family is recently discovered and prevalent member is interleukin 17. Another family is chemokine family. Chemokine families members are CCL19, CCL3, CCL2, etc. etc. And we would look at the function and their mode of action gradually in this video. So stay tuned and watch the whole video. So first we would talk about the interleukin 1 family. Now interleukin 1 family members are mostly pro-inflammatory cytokines. So IL-1 alpha, IL-1 beta, IL-18, IL-33, all of these are pro-inflammatory cytokines. Means that they give rise to swelling, redness, etc. and all the inflammatory signs. So also IL-1 family has anti-inflammatory members, which are IL-1RA, which is IL-1 receptor antagonist, IL-36 receptor antagonist, and all these things. So there are also secreted version of receptor antagonists which actually oppose the inflammatory cytokines of IL-1 family and thereby check inflammation. So here how the IL-1 receptor mediated signaling occurs. So IL-1 binds to it IL-1 receptor. IL-1 receptor is a heterodimer consisting IL receptor 1 and IL receptor ACP. Now all of these circular domain that you see are actually Ig fold. So this receptor is of a broad family known as Ig superfamily, and it's the same family member of an antibody. And at the cytoplasmic end it has an important domain which is known as the MyD88 binding domain or TIR domain. So MyD88 also is known as myeloid differentiation protein, and it also has this TIR domain. So they interact with each other and works as a scaffold. MyD88 works as an adapter or a scaffold which would allow association of TRAF6, which is a TNF receptor associated factor. And TRAF6 would activate an important kinase known as IRAK4 or IRAK6. So IRAKs are basically interleukin receptor associated kinase. IRAK does very important function. So in the cell there is another important kinase known as TAC kinase. So TAC kinase is inhibited in a complex where TAB1 and TAB2, which are TAC associated proteins or TAC binding proteins, are sequestering TAC. So this TAC kinase is not freed for further downstream function. What IRAK does is phosphorylating TAB1 and degradation of the TAB1 and TAB2 complex, and thereby freeing TAC. Once TAC is free, it can also phosphorylate IKK beta of this NF kappa beta complex. Once phosphorylated, IKK beta becomes unstable and ubiquitinated. So NF kappa beta becomes free, and NF kappa beta moves to the nucleus. In there, it gives rise to transcription of specific genes, including cyclin D, which give rise to cell division. So once this particular signaling cascade has a proliferative role. Also, it gives rise to several pro-inflammatory molecules, which ultimately give rise to the inflammatory signs and symptoms. So overall functions of IL-1. So IL-1 is a prevalent pro-inflammatory cytokine. So definitely it would increase vascular permeability. It also modulates adaptive immune response. That means immune response of a B cell and a T cell. It definitely force the vascular surface to express several adhesive molecules, which in turn slows down specific immune cells such as neutrophils in the vascular surface. That helps in neutrophil extravasation in the injury site. Also, IL-1 forces the liver to form specific acute phase proteins, which are in turn important for inflammation. Now we would come to hematoprotein family. Now hematoprotein family name come from the hematopoietic pluripotent stem cell, so they are all important. Earlier they are thought they are important for this hematopoiesis function. Now hematopoietin family has important members. One of the most important member is interleukin 2. And interleukin 2 has a very important role in the immune system. So the basic characteristics of this hematopoietin family is they has a 4-bundle helix. So this is the structure of the 4-helix bundle that hematopoietin family cytokines possess. Now let's say this T cell is actually activated by a peptide that is presented on a class 2 MHC by a macrophage or any other antigen presenting cell. Now this would ultimately give rise to 2 signals for differentiation or proliferation of that particular T cell, and the third signal is actually interleukin 2. The interleukin 2 actually works in autocrine fashion and works on the same T cell, and that helps the T cell to undergo cell division, and that ultimately the naive T cell then would proliferate and form a pool of memory T cells and a pool of effector T cell. Now these IL-2 receptors appears in 3 conformation. In a high affinity conformation where IL-2 receptor has 3 chains, namely alpha, beta and gamma. It can also appear in an intermediate affinity form where it has beta and gamma chain, and also it can only appear in a singular form of alpha chain, which is a low affinity receptor. Now here is a list where these receptors are found, where these intermediate affinity receptors are found in mostly resting T cells and natural killer cells, and mostly the high affinity receptors and the low affinity receptors are found on the surface of activated CD8 positive T cells and CD4 positive T cells. Now the question is, if activated CD8 positive T cell and the CD4 positive T cells has this high affinity receptor, then what is the function of the low affinity receptors? It turns out people speculate that low affinity receptor could be useful to sequester IL-2 on the surface of these cells and then later pass that onto the high affinity receptor. However, its actual mode of action is not totally clear. Other family members include GCSF, GM-CSF, and IL-3, IL-5, etc. So they also have 2 different configurations. One is a low affinity receptor configuration, another is a high affinity receptor configuration. Why do they have both sort of receptor? It's still unclear. But whatever may be the reason is, the overall functionality of the hematopoietin family of cytokines are, first of all, hematopoietic pluripotent stem cell differentiation and proliferation, neutrophil proliferation, basophil degranulation and basophil proliferation, and also they help in megakaryocyte and monocyte differentiation. Now we would talk about interferons. As the name suggests, interferons means that would interfere with some processes. And I have a detailed video describing the interferons and their mode of action. So if you haven't watched this video, you can watch the video in my immunology playlist. Now, interferon has 3 families: interferon class 1, interferon class 2, and interferon class 3. Interferon class 1 comprise of interferon alpha and interferon beta. whereas class 2 has interferon gamma and interleukin 10. Interferon class 3 is a newly discovered family comprising of interferon lambda. Now, interferon alpha and beta help in T helper cell differentiation, switching the naive T helper cells fate toward Th1 CD4 positive T cell. Now, interferon alpha and beta also works like alarming signals. So, macrophages and dendritic cells secrete interferon alpha and interferon beta, which in turn alarm the nearby cells to come to that particular location. Now, interferon gamma has a huge role against viral protection. So, it can lead to, so what happens is, so when a cell is killed by a CD8 positive T cells, the dead cell secretes some molecules. And these molecules turned out to be interferons. Now, what happens is, interferons, which are secreted from a viral infected cell, that bind to an interferon receptor. Interferon receptor signaling give rise to specific gene transcription, which ultimately produce antiviral molecules. And that in turn save the neighboring cells from further virus infection. So what are these antiviral molecules produced by interferon signaling? So let's say the interferon, one of the most important molecules that are synthesized due to the interferon signaling in the neighboring cells is protein kinase R. Protein kinase R actually binds to viral dsDNA and inhibits its translation. Also, protein kinase R, being a kinase, can phosphorylate initiation factor 2 alpha and indirectly inhibit translation. Second strategy is forming 2,5 oligoasynthase, which actually binds with viral dsRNA and make it degraded by RNA's L activity. So the mRNA is getting degraded and no viral protein is formed. In the third strategy, what happens is MX protein get polymerized and they inhibit the viral coat assembly, such that several life processes of the virus could be interfered by the function of these interferons, and hence their name is interferon. Next family is chemokine family. Now, chemokine family works like a chemical messenger and they form chemical gradient. For example, here is an injury site and the chemokines are secreted by several cells which are in this injury site, and the chemokine forms a gradient, a high to low gradient from where the infection happens to a distant part. So any molecule, any specific immune cells which is migrating along the blood vessels would identify the site of injury or the site of pathogen invasion by migrating along through a low to high chemokine gradient. So in other words, they help in cell migration and immune cell navigation sort of thing. So one of the most important thing that they do is they increase the surface expression of the adhesive molecules on both the surface of the specific neutrophil or basophil inside the blood vessel and also the capillary endothelial cells of the blood vessels. So that helps the particular immune cell to stop and roll slowly, such that the diapedesis or neutrophil or basophil extravasation could occur and it can reach the injury site. Now, all these chemokine receptors are actually G protein coupled receptors, and they can work in multiple different signaling pathways. They can activate one of the most important signaling pathways which regulates growth and cell division, which is the mTOR signaling pathway via protein kinase C. They can also activate RAS, and RAS can give rise to downstream effect by MAP kinase signaling pathway. All these things works via AP1 family transcription factors, which give rise to particular proliferative genes such as c-MYC, cyclin D1, also some genes like anti-apoptotic genes like BCL-XL and BCL2, etc., etc. Also, phospholipase C beta mediated signaling can release NF-kappa beta to the nucleus, And first of all, since these chemokine help in cell migration, so cell migration requires a lot of cytoskeletal rearrangement. And the cytoskeletal rearrangement and then cell migration could occur because a chemokine mediated signaling activates a lot of small GTPases known as Rho, CD42 RAC. All these things are known to activate the actin polymerization, and thus cell migration is regulated by chemokine signaling. Now we come to, and all these kind of signaling would ultimately lead to either cell proliferation, cell migration, or cell differentiation. Now we would come to TNF family or tumor necrosis family. Tumor necrosis family has TNF alpha and TNF beta. All are pro-inflammatory, and also they are actually apoptotic. But not only they are apoptotic, they can sometimes give rise to survival signal as well. For example, TNF alpha and TNF beta that are secreted from macrophages are actually giving rise to inflammatory signal, whereas BAFF and APRIL has certain important role in B cell development. And also fast ligand are actually expressed by cytotoxic T cells and NK cells, and they have certain roles in actually killing the target cell. So here is the TNF alpha family receptor. Now TNF alpha receptor binds with tumor necrosis factor. So tumor necrosis factor binds to that, and also they have a cytoplasmic domain known as death domain. Now TRAD is also an adapter molecule having the death domain. So death domain and death domain interaction makes a solid scaffold where other signaling molecules can come on and interact. So other signaling molecules known as RIP and TRAF2 interacts with the TRAD, and ultimately it activates a particular molecule known as FAD, which is fast associated ligand with death domain. Now FAD actually converts pro-caspase 8 to caspase 8, which is a active form of caspase 8. Now once caspase 8 is in active form, it can cleave procaspase 3 to a caspase 3. Now caspase 3 is a executor caspase. Caspase 3 can further cleave downstream a lot of signaling molecules, a lot of proteases, which would ultimately cleave the nucleic acid in the nucleus, the cytoskeletal framework of the cell. Ultimately, it would program the death of the cell. But there is another format of signaling used by the same receptor. So sometimes TNF alpha and TNF alpha receptor mediated signaling could lead to survival. In that case, the signaling happens in a little bit different way. So definitely TRAD, RIP and TRAF, etc. interacts with the death domain of the TNF receptor. But the key difference is when cellular IAP and CIAP2 and LUBAC binds to this complex. It don't allow the RIP to fall down from the, so it allows the RIP to fall down from the complex. And what happens is RIP frees TAC kinase from its inhibitors, which are TAB2 and TAB1. Once TAC is activated, TAC phosphorylates several target proteins in the MAP kinase signaling pathway, such as ARC, MEK, etc. And also, TAC phosphorylates IKK beta in the NF kappa beta signaling pathway, releasing NF kappa beta and such that NF kappa beta can migrate to the nucleus. So once NF kappa beta pathway is activated, one of the most important downstream molecule is known as C-flip. Now C-flip is an inhibitor of caspase 8. So it can suppress caspase 8 and actually check the apoptotic pathways, and also MAP kinase pathway and NF kappa beta pathway can ultimately give rise to a lot of proliferative gene and anti-apoptotic genes such as BCL2, BCLXL, and cell division related genes like cyclin D, cyclin E, etc. So all these things can give rise to proliferation. So it depends whether these TNF alpha mediated signaling would either give rise to death or proliferation, depends which intermediate signaling players they're using. Now we come to the clinical use of these cytokines. Now Enbrel, which is actually TNF receptor and IgG chimera, is widely used in treatment of rheumatoid arthritis. Then Humira, which is a monoclonal antibody against TNF receptor, is used for treatment of rheumatoid arthritis and Crohn's disease. Roferon A, which is interferon alpha 2A, is used for treatment of hepatitis B, hairy cell leukemia, Kaposi's sarcoma, and hepatitis C. Avonex, which is interferon beta A, is used to treat multiple sclerosis. So these days, cytokines, especially interferons, are used for several clinical purposes. So that was my video on cytokines. If you like my video, give it a quick thumbs up. Don't forget to like, share, and subscribe. Thank you.